Uh, Good afternoon to everyone. Today we are going to learn some basics about three important molecular events that happens inside the cell, in a cell. This might happen during the cell cycle, maybe in the G1 phase, you have to remember synthetic phase, G2 phase, and cell division. So we will discuss three important molecular events that happens in a cell because these events will have to happen in order to adhere to the principle of central dogma. That means the central dogma tells, this was explained by Watson. What he told was that the DNA, that is the genetic material, is responsible to produce the proteins, and these proteins will in turn carry the inheritance. That means any character we see today in any organism, that is transferred from DNA to RNA, and RNA will become protein, and that protein changes into organs, and then inheritance is possible. Sometimes RNA may also help in DNA, and the DNA becomes RNA. This kind of happening where the inheritance takes place via DNA was considered to be central dogma. I think it still follows. Well, we will not go to details of those things, but we will try to study three important things so that happens during this particular thing. Why we have to remember the cell cycle here? This is cell cycle. Why should we remember that? Because in the G1 phase, most of the cellular materials will duplicate, except DNA. So almost all the things they will duplicate, and the DNA especially duplicates in this S phase. And then in the G2 phase, they check the correctness of the whole thing, and then they go to cell division, that is, new cells are formed. These new cells, when they grow, they will have the design of the parent. That for that particular thing, there must be duplicate of everything, and all the characters will have to be carried to the parents. How that is done? That is because of the genetic code. That is what the central dogma is. In central dogma, it says that all the characters of the parent will be transferred to the offspring via genetic material, and that happens because DNA will pass it on to messenger RNA, and messenger RNA will take it on to a place called ribosome, and where the ribosomes will synthesize proteins, and proteins will form the tissues, and tissues will form the organs will form the organ systems. So this type of a complicated structure thing was explained by Watson as central dogma. So we are going to examine that. We will not go much details to all these things. So what we say here, in the S phase, especially the chromatin material divides into a large number of pieces called chromosomes. Each species will have a definite set of chromosomes. One from the one parent, another from the another parent, like that. There will be two sets of these chromosomes here. Two strands will be there, or two types will be there. So in this case, I have shown 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This is only a hypothetical case I have taken. So that means here the chromatin divides into a definite number of segments called the chromosomes, and these chromosomes carry all the characteristics from the two parents, one from the father, one from the mother, they will carry, and these chromosomes in turn they contain genes, and these genes will be passed on to the offspring, and these genes will produce the required proteins. So this is how the entire thing happens. We will not go to details of all these things. So we will straight away take the three molecular events that happens in a cell to carry on the central dogma, to make sure central dogma works, that happens. The three important molecular events are, one is called the DNA replication. Second is the DNA transcription. Third is called translation. So we will remove this word DNA. So we remember three molecular events: the replication of DNA, transcription, what is transcription we will know shortly, and then translation. These two happens, that is 1 and 2. These two will happen in the nucleus, and translation happens in the cytosol or cytoplasm. So these are the three major molecular events that are responsible to transfer the hereditary characters from parent to the offspring. We shall see what they are. Right now I am going to write a small diagram which is very familiar to you, perhaps. I hope this structure you know very well. This is a double helix of DNA molecule. A small portion of the DNA molecule is taken from the chromosome. It's a chromosomal DNA, and this is tightly coiled, and it may be tightly coiled here like that. It will be tightly coiled in the things, and many proteins and enzymes will work upon that, and then they will extend it. They will try to see that this coils are uncoiled. Uncoiling, you call it, uncoiling takes place, and then you get this kind of a long stretch of a DNA, that is DNA double strand. We can see DNA double strand, we can see. I'm giving less information, but we try to understand this thing. This will be DNA double strand. This is how the DNA is theorized to appear when it is uncoiled. You know, it will all contain, DNA contains the nucleotides. Nucleotides contain the phosphate, a sugar, and then a base. This will be one nucleotide. Like that, if you just shorten that, there will be a large number of nucleotides in a DNA molecule in a DNA piece, and I will use another color so that it will be easy for us to explain. So there will be the nucleotides on both these strands, and these they will be linked or connected at the place by means of hydrogen bonds. Like that it will be there throughout. So all these things will be connected. So this is the basic thing we require to understand about the DNA replication. So a DNA strand will be there, and this DNA strand may be a small segment here, maybe a small segment, and that segment may be a gene. If that gene is taken and extended, it will show a large number of specific sequences of nucleotides. So this has to be now doubled, because during the synthetic phase, this kind of DNA will now, I will just make a short thing to explain to you, then we will explain. Supposing you take only this portion of it, and we say this becomes replication takes place. What do I mean by that? So let's say this is the parental DNA strand which I am writing here. This is the strand here, I have written here, and this is another one. I will use a different color here. Here also I use a different color so that we know what we are doing. Right, so this is the one more strand here. So this portion I am writing here. So when you examine that two pieces, you find these things have the nucleotides. That's one nucleotide, another one, another one, another one. I have taken only 4. Like that, each gene may contain hundreds and thousands, sometimes millions of nucleotides. And to this, one more nucleotide is added here. I have taken 4. What I do, I will try to change the colors also so that we will understand very clearly. Now this is okay. This one I will change the color of this. I will change it. That is one color, one type. And then this I will change it to another color. We have now 1, 2, 3 colors are there. And then I will try to write this, and I will change this color to yellow one, and then like this. So there are 4 basic types of nucleotides connected by means of hydrogen bonds. They will be connected here. Those details will not go. So if this type of a DNA has to be replicated, that is one copy, and we have to make the same copy. How it happens, quickly we will study here, then we go to the next one. This is one strand. It will be taken care by many enzymes. At least about 5 to 6 types of enzymes will be there. These enzymes, and also there will be proteins. Some proteins will be there. So these proteins and enzymes will now break this one, and they separate the strands. When they separate the strand, one of the strand may look like that. Now it contains, you see, one of the base here. The second one is here, and the third one is here, and the fourth one, which I have written here in the white, that will also be there. So this is one strand, one of it is separated, and this one, let's say this is 2 and this is 1. So 1 is here, and the second one also will be separated like that by enzymes. I told you the action of enzymes. This again, and this color I am writing here, and the green I am writing here, and another yellow I am writing here. Supposing this is the way they separate, and then what happens? The there are if you are already studied the DNA molecule, you know very well. There will be 3' end and 5' end or 5' end, and this will be 5' end and 3' end, and same thing here. This will be 3' end and this will be 5' end, like that, and this will be 3 and this will be 5. So if you know these things, it will be easy for you to understand. If not, don't worry, we'll see. Now what happens is the enzymes pick up materials. Yes, one is called polymerase, and that will bring the new opposite type here. For this there was a blue, so they bring a blue one here. And there was a yellow one here, and they bring a yellow one here. And that was a green one here. Green is there. And then another blue was there, that will be there. And the basic backbone is also made. That backbone is also made like that. That is one copy. And similarly for this one, what they do, they will again start collecting. For this one, blue, there was again a white one. And for this yellow, there was a blue. And for this green, there was a violet color. And for this blue, there was a white one. And the backbone comes here. This is a copy. This is a replica. So now these two are exactly the same as the parent was. This is what happened during the replication. That means on the chromosomal DNA undergoes many changes. The strand separates, it's called separates, or I will come to you. At this point, enzyme will work, and they make a fork like that, and they completely remove, and what they do, they start separating, and then they become completely on a very, very far away structure, and that is called the fork. Now fork formation takes place. And this is taken by means of an enzyme. There is an enzyme here, and that enzyme is going to work and produce the fork. Okay, and we call that enzyme as helicase. What I do, I will try to rub off these things. All these things I will rub off now. We will write it again. I think by this time you must have understood that the original DNA, which was in the form of a tight coil, has been made loosen and become a long stretch of double helix. And that long stretch of double helix will now be the template to produce two new strands to form replicas. Now we will come to the same diagram here. We say this is called the fork. What we call the replicating fork. Replication fork has come up here. Now this particular enzyme is called helicase. I told you many enzymes will work. We will not write the names. Scientists have given different names for them. At least about 10 or 12 enzymes have been identified in this one. I am teaching for the classes for maybe 9th and 10th and 11th area. Therefore, I am not willing to give more complicated names in this, except a few names. That's one enzyme. And then what happened is, here we have 4 bases. One base, second one, third one, and the fourth one. Now these bases will repeat. Now the names for these bases are, one of them is called adenine, let's say adenine, and then this will be thymine, and then this will be, take the color, let's say this will be cytosine, and that may be guanine. So this is the four pair. To this, in this strand, in this opposite strand, exactly different one will be there. Adenine always along this adenine, let's say the adenine is here, and for this adenine, adenine always goes with the thymine base. There is thymine here, and here we have thymine. For this thymine, again adenine will be here, and then here we have the cytosine. For cytosine, always there will be guanine. There will be cytosine will go with the guanine, and then we have the guanine. Guanine will go with the cytosine. Right. This is the thymine. Thymine will go with the adenine. I have given the color adenine, and then cytosine will go. This will be thymine. Correct. This will be say adenine, guanine, and then that will be blue color. Okay, I made a small mistake, corrected that. So now these are the base pairs. Like that, you may find a large number of nuclear bases here, which I am writing it randomly so that there is no confusion for us for understanding the basic structures. So all this will be taken care like that. So that means the helicase will attach to a point where it starts making the helix into a fork-like structure, so that the inner DNA structure is opened up for the replication to happen. That is the very first thing that happens. Then there are many events. We will not go into details of all those details. What we say, this is the we will say this is 3 dash or 3 prime end. This is the 5 prime end, and the same thing will be written here. For this, there will be 5 prime end, and this will be 3 prime end. This is what it is. What they are, we need not worry too much about it. Now, at this part of the time, the many enzymes will start taking part on that one. Now, one of the things that will happen is they will identify a spot. One spot is identified, that is the origin spot. Who identifies? Again, it's all due to the work of enzymes. And then to that spot, one small structure called RNA primer. RNA primer attaches itself here. That's required. It attaches, and then when once it attaches, immediately at this point there will be a DNA polymerase. One more enzyme comes, and that enzyme is called DNA polymerase. So there are so many things they call it alpha, beta, gamma, epsilon, and all these things. We will not go to that. We will only say DNA polymerase has come, and we shall say DNA polymerase 3 has taken place here. And this one will now initiate the formation of the new chain. What it does is, it starts collecting the nucleotides, and then it keeps on adding from here it starts. This is the 3 prime end, and when it is 3 prime end, this will be the 5 prime for this one, 5 prime for this. So from 5 prime end, it keeps on adding the nucleotides like that. Now this particular strand will get the backbone. So a new strand is formed on this particular thing, and this is a continuous thing, and that continuous strand is called leading strand. Okay, leading strand. So this continues like that. We will come to that later on. Whereas this strand, again, it has to start from somewhere from here. And what happens is, when it again opens, and when it opens, when this completed, this now attaches the helicase will attach here, and then it again further it wide opens the DNA fork. The fork becomes much more bigger than what it was. DNA fork is again open, and again here we have the structures. The molecules, nucleotides are there. The bases are there. So like that, and this keeps on growing. As this is happening, this particular portion looks to be lagging behind, and therefore this strand is called lagging strand. Now a very unique thing happens here, which was maybe identified by one Ogasaki, a scientist. So he said that instead of coming here, he says somewhere here, somewhere in the middle or in any part, the one RNA primer, maybe RNA primer here, RNA primers will attach, and then they start making the new DNA, adding nucleotides to this one, and they add nucleotides, and then when the nucleotides are added, they will again have the new backbone is also formed. Like that, here also the same thing happen. Nucleotides keep on adding here, go on, and then the backbone is again formed. Like this, there will be discontinuous formation of the DNA molecule in one side. That is in the lagging strand. In the leading strand, there will be a continuous strand that goes on. As I say, this helicase joins here, opens forks here, like that it keeps on doing it. If you look at the DNA, maybe there will be this is an imagination that we can do. There will be DNA stranded like that, and we may find a large number of small bubble-like structures where the DNA replication may be happening. They are called DNA bubbles, where all these events what I explained to you may be happening here. And then at the end, when it reaches the end, then there will be one strand, very long strand, that has come from 3' to 3' and that goes on. That is the reading strand. It reads on that one. And then the other one will have small segments of DNA. Those segments are called Wakasage segments. And these segments will have to be joined now. To do that, there is another enzyme that comes to play, and that enzyme is called ligase. So now ligase comes here, attaches, remove these things, and then they will patch it up, add up the sequencing material, and then that also becomes a long strand. So as these things will be happening, we will be getting 2 strands made from one DNA molecule. They are the 2 daughter strands. So like that, the replication takes place. I will just try to make a small summary here for you to understand. We said that DNA molecule will be tightly coiled, and that coiled starts opening with the help of the enzyme from the proteins, and then it ultimately comes into a structure where double helix will be very clearly seen. At some point of the origin of the DNA molecule, there will be enzymes and proteins will attach to that one. They are called binders and all these things. I have not come details to that one. We'll just make a beginning. And at one point of attachment, there will be an enzyme called enzyme, and that enzyme will helicase, called helicase. It makes the DNA helix to separate to form a long fork, and that fork will be having on two sides the DNA bases or nucleotide bases like adenine, thymine, cytosine and guanine, and then at one point of time a primer called the RNA primer. It's an RNA molecule of 3 or 5 segments. It has 5 bases. It will come and attach. When it attaches, immediately to that there will be another molecule which we call the DNA polymerase. DNA polymerase, and the DNA polymerase attaches, and then that starts adding new nucleotides to the strand, and the strand keeps on growing from 5' end to the 3' end. It is continuous, and therefore that strand is called the leading strand. And then another strand where at the point where the helicase will join, and that will be left free. That means the duplication, replication happens if this happens in one direction like that, this strand will have to form in the opposite direction. So it is bidirectional. Because of the bidirectional way of making the new strands, there will be small breaks, and those breaks are called the Wakasagi segments. It is called discontinuous formation, and that strand is called the lagging strand. Then the other enzymes will take over. They come and they remove the RNA primers, and ligase will come and join that, and ultimately two similar daughter strands are formed having the equal amount of DNA in quality and inequality, and such a kind of activity takes place in the nucleus, and that is called replication. Now I will just tell you, replication is a very complicated process that happens both in the prokaryotes and the eukaryotes, but we are interested in the eukaryote one. Prokaryote means the organisms which does not contain the nuclear material. Nuclear material does not have the envelope like bacterial cell. Eukaryotes have a new or the correct nucleus, or the U means true nucleus, having the outer nuclear membrane. So in those, we have now taken up the replication part in nucleate. So we found that it is a complicated process. It will a large number of enzymes will take part in that, proteins will take part in that, and then there will be nucleoplasm will contain the bases in the form of nucleotides. New nucleotides are added, and ultimately we get 2 copies of the DNA, one DNA strand, which is exactly like the same, both in quality and in quantity of the number of base pairs. It will be the same, and thus the DNA replication takes place in the cell cycle, especially in the synthetic phase. If there are 23 pairs of chromosomes, there will be 46 single chromosomes in the cell at that time. And when the cell enters into the mitotic cell division type, both the cell division, that is the nuclear material, will separate, and they will again join, all the chromosomes will join with one another, they form the chromatin, and then they establish themselves into nucleus. This is the way how the duplication of or what we call the replication takes place in the eukaryotic cells. Thank you very much.